need to do when I look and sit down to evaluate a person's genetic analysis is by this time I've seen several of them. And so I no longer use the beginning of the Gene SNP report that is received from the lab, that goes over genetics 101, that goes over how to read the report to identify the things that I would want my patient to know. When I sit down with a patient, however, I make sure that they see that information, that they see the genetics 101, that they see that there are ways that they could read this report, that there are ways that they could get their own interpretation or their own results and identify for them where they're going to make their changes or how they want to change their commitment. The one aspect of this report that everyone focuses on is the obesity scale. There is on the Gene SNP report your general obesity index, and it gives you an obesity score from 1 to 10. And everyone is upset when that number is above 5. Let's say it's 7.9, like the report in front of me right now. Immediately, two things will flash through the patient's mind when they see that obesity score. They're either going to say, that's why, that's it, it's not my fault, or they're going to say, this doesn't make any sense to me. I question the results because I'm not obese. I'm not overweight. How could I have a high obesity score? So let's just get over that right now. Genetic analysis, nutrigenomic analysis is not a diagnostic test. It does not tell you that you have anything. It tells you that you have genes that may behave in a certain fashion, and that we as practitioners or people that are talking about nutrigenomic analysis can change the way a gene behaves with the gene's environment. So having a high obesity score, like 7.9, does not mean that the person is overweight. It means that they have a larger number, by percentage, of obesity SNPs. If the patient in front of us is not overweight, they're not obese, and they have not struggled with weight management factors, then we know that those genes are more than likely not turned on in the individual. What I would like to make sure that people understand is that the Gene SNP DNA analysis goes well beyond an obesity score. Obesity SNPs are not just a risk factor for obesity, but also a risk factor for where obesity is a primary concern, like blood sugar issues, like longevity, like gut health, like cardiovascular health, like fertility. So we want to now expand on this idea of this obesity score and think if there's a larger than average number of obesity SNPs, it may mean that there's a larger than number of health risk factors that we need to assess in this patient that are related to obesity. But want to be sure that everyone understands having a high obesity score is neither a sentence of obesity, nor is it should be the qualifier for whether or not this test is valid or gives valid results. It's simply a question of these are genes that have the potential to be turned on and cause obesity. And I would just like to take us one step further and say if they can be a risk factor in obesity, then they are can easily be considered a risk factor in heart disease, endocrine dysfunction, sleep issues, gut health, and the like, everything that obesity has to do with. But from there, you want to be able to manage that expectation with a patient and be able to dialogue with them. And so that's how I use the beginning of the Gene SNP reports. Okay, so from here, we get into the results of the Gene SNP report on page 9. At the top of each one of the report, or each page of the report, you're going to continue to see this obesity score. Part of that has to do with the beginning of the test talks about obesity. One thing I think is important to note when it comes to reading the Gene SNP report is that the health practitioner guide is not in the same order as the report. The report is separated into panels: an obesity panel, a fitness panel, a food, a macronutrient panel, or a vitamin utilization panel. We wanted, with our health practitioner guide, to go beyond that and give you more clinical references. And so we put the Gene SNP DNA Analysis Health Practitioner Guide into a different order, or different segments. So we put it into genes associated with behavior, for example, genes that are associated with lipid metabolism, for example, genes that are associated with weight gain and obesity. This is the panel that gives you insight not just into the obesity SNPs, but maybe some of the struggles that people will have to lose weight, going beyond the foods that they eat, and giving you insight into sleep habits or mental emotional habits as well. An example in this gene guide is information about individuals that will eat very strictly and lose their weight, but if they don't stay strict with their gene diet or their supplement program, they have a higher than normal tendency to gain that weight back. Well, on the flip side of weight gain and obesity, there's also a panel that goes into weight loss and fitness and gives you some insight into that. A panel on antioxidants and immunity that gives you some insight as a practitioner into detoxification pathways. We do get into sharing some information with the patient report when we talk about nutrient metabolism. The difference between these two reports will be when we look at nutrient metabolism with the health practitioner guide versus the patient guide, which is what's received from the lab, that we're going to go into more of the clinical manifestations of vitamin D metabolism, of carotenoid utilization, or alternatives to carotenoids if, for example, you have a smoker as a patient. That's the type of information that will be different from the patient guide to the practitioner guide that was developed by physicians for the help or to help practitioners interpret information. And then we go into a panel that is not explored in the patient guide that talks about methylation pathways or methylation SNPs. So the first thing I'd want people to know when they evaluate the patient guide versus the health practitioner guide is that they're not in the same order. The patient guide is put into the panels that the laboratory chose. The health practitioner guide is put into segments of health that are relevant to the practitioner and the clinical presentation of their patient. And so that's my first recommendation when sitting down with the report, is to identify some of those differences so that you can see when and where you're going to use one guide over the other. So my first step, once I have recognized that the information that's in the patient guide is not only designed differently in the health practitioner guide, that is in a different order than the practitioner guide, is to also appreciate that the patient report that comes from the lab that gives us our Gene SNP results may also have what some practitioners or patients may consider contradictory information. You may have a Gene SNP polymorphism that is red or green in the patient guide, but bears a different color in the health practitioner guide. The reason why they will have different colors from guide to guide is because in the health practitioner guide, we evaluated some of the clinical manifestations, not just the obesity manifestations. So it's possible for someone to have a green color allele in the patient guide because it is not a risk factor for obesity, or it is not a risk factor for poor beta carotene utilization, just as an example. But when we look at the health practitioner guide, it may be a risk factor for mitochondrial function. It may be a risk factor for blood pressure. And so it would be red in the health practitioner guide because there's an additional clinical manifestation outside of the more generic or patient profiled Gene SNP analysis that comes from the lab. From there, I need to then appreciate what alleles do my patients have, what SNPs may be at risk, and where do I want to put my focus with the patient. Undoubtedly, you want to put the focus on gene SNP interpretation on some of the primary concerns that your patient has, some of the presenting complaints that a patient has. You would then, from the next step, or what you want to move into next, after saying if my patient struggles with sleep, or if my patient struggles with energy, or if cardiovascular health concerns are at the top of my patient's list, I definitely want to go through my report and identify which SNPs are going to help me answer those questions so that I can see what proportion of those gene polymorphisms fall into that basket of risk factor for cardiovascular disease, or risk factor for fatigue, risk factor for energy, and the like. So from there, my next step is then going to evaluate my gene polymorphism report next to the health practitioner guide via the cheat sheet, so that I can see how many across the board deleterious alleles does my patient have, and then pair that information with my patient presentation. Is this a concern or is this not a concern? So getting straight to that, I need to take the cheat sheet, take the patient report that comes from the lab, and start filling in my results. I'm going to start on page 9, and I'm going to start on the first page of my cheat sheet. That page starts with the FAME2 variant. On the cheat sheet, it'll tell me the genetic variation that I'm looking for, and then we'll go into some information about product utilization, but right now I only want to use the first 4 columns, the gene variant and the allele or the genetic variation result. So going from the patient guide to my cheat sheet, I'm just simply going to fill in the information. If my first allele is FAME2, in this case, it's a GG variant. I'm going to mark that down on my cheat sheet, and I'm just going to go through each one of them and mark my alleles. By doing this, I'm able to find out how many are yellow, how many are red, how many are green. I can now see, is this a gene result that's going to have a lot of spaces for intervention, or are we going to look at just a handful of gene variations? And I'm making that decision simply based on the color of the gene. Someone else can definitely do this part of the gene interpretation. A staff member that we assign can fill in the information. There's nothing clinical about this part of the interpretation. It's filling in the blanks, filling in boxes. You just want to make sure that you don't lose your place, or that you come back to the report and make sure that you're in that you're in the right place, because the information that we put together for interpretation is in while it's in the same order, it may have different colors than what you see in the lab report. For that reason, you want to take those little visual cues and stop and make sure that you're on the right SNP. But absolutely, this is not critical that the practitioner do. A staff member could easily do this for them. One thing is, we think about going through this, and we identify, because we've seen more than one, and we identify that there are certain SNPs that are associated with a common patient presentation, high blood pressure, for example. It is possible, as you go through this, to find that there'll be one gene SNP polymorphism that indicates a risk for high blood pressure, and to see another allele that is a risk for high blood pressure, but see one indicate on the patient report that blood pressure is not a concern, and then in a different place on the report say that blood pressure is a concern. That is not a contradictory finding. That finding says that due to a different reason, blood sugar may be the reason why somebody has elevated blood pressure, while salt or water retention may be another reason why someone has elevated blood pressure. It is possible to not have one blood pressure SNP, but have the other. Blood pressure is still a risk factor, but it's not a double whammy. To do that, I do think it is smart. What you do need to make sure that you do is that you have read this report before the patient is in front of you. It's really important when you do Gene SNP DNA analysis that you've opened up the kit before you take the swab, that you know what steps you need to take, and that you've already explained to your patient that they cannot have eaten anything or drunk anything for at least 4 hours. That you're not going to draw a sample on a Friday, for example, because this needs to go in the mail, and there may be an issue with Saturday pickups, which means you've spent, of the 7 days you have to get this to the lab, you spent 3 of them over the weekend because it wasn't picked up on the Friday. It may not be picked up on a Saturday. So for that reason, I think it's important to, before you even do your first Gene SNP analysis, that you've read through the literature, that you've read through each one of the reports, that you're familiar with the tools, that you've opened up a kit, and that you've become familiar with the swab, with the handling containers, with the stickers, and of the 4 stickers, where do they go? 1 on each of the swabs, 1 on the outside of the tube, and the last one is part of your patient chart, or that information transferred in some fashion to your electronic chart. Those are the steps that definitely become kind of rote repetition after you've done more than one. But you'll find yourself frustrated if the first time you do any of these SNPs is when someone is watching you learn how to use the Gene SNP DNA analysis, for sure.